Okay, so in this video I'm going to demonstrate how to prepare smears to make stainings. So a smear is a thin film that goes into a glass slide, which we have here, and then the first step, of course, is to turn on your Bunsen burner so that you can fix the smear with heat. There are two ways to fix it, with alcohol or with a flame. And you can make a thin film either with broth, which is very easy, or with slant, which is solid media. So first, let's look at how to make a smear with broth. So what you do with your glass slide is that you identify your glass slide here with the name of your bacteria or your microorganism of interest, the initials, and the date. So in the broth, I have a bacteria, it's called Enterococcus fecalis. So for abbreviation, it will be E. fecalis. And then your initials and the date. After you do that, you will be using a loop, inoculating loop. You will sterilize it, as you know, 45-degree angle in the middle of the flame. Put it on one side, let it to cool. And the reason for let it cool before grabbing the sample is if you introduce your loop when the loop is hot into the media, that is going to kill the bacteria. And then the area where you're going to put your smear is going to be here in the center of the slide. And you work here in the sterile area. So as I mentioned before, working with broth is very easy because the bacteria is suspended, and you just grab a loopful, one or two, and then put it in the middle, and then with circular motion you will distribute it to make it much even than just leaving the puddle in there. Now in broth, bacteria tends to go into the bottom when it's static, so you just have to shake it a little bit like this to suspend a little bit better the bacteria from the bottom into the middle to make a homogeneous mixture. And then I think right now it's already cool, so I'm going to just sterilize this ending of the tube, introduce my loop in the middle of the tube with media, sterilize it again, put it here in the rack, and then the loop, you can see it with the bacteria there. You just put it in here in the middle, and then you just spread it. After you spread it, you let it dry, and then once it's dry, you can heat fix. So since I'm going to make with solid media as well as smear, I'm going to show you, while this one dries, I'm going to show you how to make a smear with solid media. So now that this one is sterile, what I'm going to do is grab my distilled water bottle, add a loopful of water into the loop by squirting the water in there, and as you can see, now I have water in here. So that one will go into a new glass slide, but first let me do it again because I haven't put the identification in here. So now remember that once you sterilize your loop, you don't put it here in the countertop, which I already cleaned with the disinfectant. So you just put it there in the Erlenmeyer flask. So now the one that I'm going to make, it's a gram positive, it's S. aureus. This is in a slant. So I'm going to put here S. aureus, and then my initials and the date. Again, it goes in one ending. Sometimes, just to make sure that you know where your smear goes, I mean, you can put like a couple of lines in the bottom where the slide has this part, and then on the top you're going to add the bacterial sample, or you can make a circle. But I'm not going to do it this time. So again, I'm going to sterilize my loop again. And the reason for this is, although I'm going to add water after this, it's just you don't know if these ones were previously sterilized. Okay, so you don't want to introduce any bacteria that is not from your strain into there. And then in here, I mean, you see that I didn't wait so much for this to cool off. I mean, it's going to cool off with the water, but you can see a drop. Depending on how much bacteria you grab is how much water you're going to add, but typically you add 1 or 2 loopfuls, and you don't grab as much bacteria to prevent having a thick smear. Smears, they're supposed to be, you can get closer, they're supposed to be thin, and you can see it guys there. It's just 2 loopfuls of water. So I'm going to sterilize again my loop, and now I'm going to let it cool. And let me tell you, while this one cools, some mistakes while making a smear. Not sterilizing your loop, sterilizing in the wrong way. Cool it, wing it like this. No, you shouldn't be doing this. So you always have to work close to the sterile area, and also not letting it dry. Heat fix is passing the smear twice before staining, and it has to be dry. Why? Because if you still have water and the bacteria there, the water is going to heat up really quick with the passing of the smear over the flame, and that is going to destroy the bacterial cells. If it's dry, it already lost the water. It's not going to pick up as much temperature when you heat fix like that. Now, another mistake is actually when you're working with solid media. In this case, this is a slant, grabbing too much bacteria. You don't have to see the bacteria in the loop. So you just touch with the outer edge of the loop, the bacterial sample, and put it in here. If you see a whitish film, that is too much bacteria. Okay, so again, you sterilize your ending of the tube, the cap of the tube, introduce your loop, not touching the rim of the tube, and then barely touching the bacterial sample, sterilizing your tube again. And then now that I have the bacteria there in the edge of the loop, I'm going to make a circular motion over the loopholes of water so that I can make a homogeneous sample. And then don't leave the puddle. So over there you can see kind of a whitish film, not as it's supposed to be, but if you smear it to the sides, you will be seeing a faint tint of white color there of the smear. So that's how it's supposed to look. And then once it dries, I'm going to be able to heat fix. And now, of course, any time that you grab inoculum or bacteria, you have to sterilize your loop again, 45-degree angle. And then now, since I'm not going to use it anymore, I'm going to put it in there. This one, it's already dry. So now I'm going to pass it twice quickly, like this. And this is already heat fixed. This one is almost dry as well, as you can see there. And yes, you can see a little bit of whitish color there on the smear. See how it's drying? But it's not as thick as this smear as sometimes it can be seen. But you see the opaque color in there. Okay, so now this one is ready to be heat fixed. And after this, you can stain it. That's all for this video.